Everyone, pardon the hiccups behind the scenes here or in front of the scenes, to our Fungi map, Funginar number 5 with Lyn Allison. Good evening and welcome, Fungi map members, visitors across Australia and overseas. We acknowledge the traditional custodians throughout this land and pay our respects to elders past, present and emerging. We also recognise the deep cultural and scientific knowledge of Australian fungi held for millennia by Aboriginal and Torres Strait Islander peoples, and we welcome you here today. My name is Susie Webster, and I thank you all for joining Fungi Maps Funginar lecture series. At any time during this evening's event, please feel free to submit your questions by clicking the Q&A button at the bottom of your window. And our hosts, myself, but particularly tonight, Ros Hart's going to take over, our Fungi Map president, and she will raise these questions with Lynn tonight. And there will be an interval for questions, and at the end, we will talk also. That brings us to our speaker and topic for this evening. Westgate Park and its fungi delights with Lyn Allison. Lyn is a relative newcomer to the delights of fungi, if not to conservation, teaching before joining the Democrats as a senator for Victoria for 12 years to 2008. Since then, Lyn has served on several boards of major not-for-profit organisations and on committees of management, sorry, pardon me, of another 4 advocacy and community groups, including the Friends of Westgate Park. Lyn specialises in governance and advocacy in these roles. To quote Parks Victoria, Westgate Park is a green oasis located in the heart of industrial Melbourne. The land has undergone many transformations before emerging as one of Melbourne's most visible parks. Westgate Park is a vibrant example of how a wasteland can become a flourishing ecological and recreational wetland environment. Westgate Park, a now 40-hectare space planted from scratch with 300 species indigenous to inner Melbourne. This sparked her interest in fungi. This amazing space has no less than 70 recorded fungal species, of which Lyn has identified more than half, and she says with a degree of confidence thanks to Fungi Map. So on that note, I'd like to introduce today's speaker, Lyn Allison. Take it away, Lyn.
Well, thank you, Susie. We'll just make sure this screen shares. And I open it up. Right. Okay. Well, thank you very much, Susie, for that introduction. That was very generous of you. I'm here with 2 hats on, as you can see. As Susie says, I'm on the Committee of Management of Fungi Map, but I chair Westgate Biodiversity, which is an amalgam of 2 community organisations. And so we used to be Friends of Westgate Park, and now we're Westgate Biodiversity, and that means we have a nursery as part of our organisation. So my proposition is that, first of all, that we need to recognise the important role of fungi and how important that is. And my proposition is that urban parks can help with this. And because Westgate Biodiversity is all about locally Indigenous plants, my further proposition is that Indigenous species of plants are our best chance of improving biodiversity in parklands, which is where, for the most part, most people go to open space. We love to go into the bush and up to forests and so forth, but typically we visit our parks more commonly. So I just wanted to start with the 2 organisations. They seem to me to be in parallel to some extent. So I've just given you there the main aims of the 2 organisations. I was part of the Friends of Westgate Park before I became involved in Fungi Map, and the reason I became involved in Fungi Map was because I bought a new camera and I wanted to try it out, and I went to the park. I ended up photographing all 300 species that were there. And then one year we had a lot of rain and the park was full of fungi, and I had absolutely no idea what it was. I must know these things, so I set about trying to find out what I was looking at. So I understood it a bit more, and so that drew me to FungiMap and eventually to come on the committee. So I wanted to start just with a little bit about fungi. The problem with a webinar is that you're not altogether sure how much knowledge people have. And I, of course, am not a mycologist. I did do biology in year 12, but beyond that, my learning is very much as a citizen science. And I like to call myself that because I have learned quite a lot, but I don't have the knowledge of people who have studied this field seriously. So I do think it's important that we have a way of saying to people what fungi is all about, how many there are. The estimate of 50,000 is probably grossly understated. We do know that fungi have not been mapped or studied to anything like the extent of plants and animals. But we do know, just by looking, that it's enormously diverse in colour, in shape, in form, and smell, and texture. And the thing which has fascinated me endlessly about fungi is the enormously diversified methods of spore dispersal. Dispersal, rather, not disposal, dispersal. This to me seems really fascinating. So I want to know how these fungi operate to reproduce. So as well as being diverse in how they look, there are a number of groups. So mushrooms, we're more familiar with mushrooms, but there's also birds' nests. So if you see in the bottom right corner, that's an example of a little bird's nest fungi. They are so beautiful, and they're easy to overlook because they're very tiny. And then there's jellies, and there's corals and cups and stinkhorns. And Westgate Park has all bar 2 of these categories of fungi, which I think is pretty amazing. So fungi fall into 3 main groups in terms of what they do. So they're decomposers, mutualists, or parasites. I think that's the full list. And there's another group of earth stars, puffballs, tooth fungi, boletes, and polypores. And of course, their relatives, the lichens, down in the bottom right corner. Now, I make an apology because there are a few photographs that are in this section which I don't have the name of the photographer for. So most of the photographs are mine, but the really special ones, like those ones down the right-hand column, are not. Some of them are Steve Axford, but I didn't have time today to accurately record who took them, but I will do that before the presentation goes public. Okay, just some fungi fun facts. Fungi evolved 1.5 billion years ago. That's a very long time ago. And globally, we understand there to be up to 3.8 million species, highly diverse. They're found everywhere, so they're in all ecosystems worldwide. Genetically closer to animals than plants, which is interesting because so often fungi are grouped with plants as if they were plants, but we now know that's not the case. And 25% of the world's biomass is fungal. Now, one of the things that FungiMap has done over the last few years is try and make sure that we don't ignore fungi as part of our biodiversity. And so we've spent quite a bit of time going through government documents, state of the environment reports, that kind of thing, where government describes and talks about its natural world. And so often, as this one, the Victorian state government one that's on the left-hand side here, talks about native plants and animals. So flora and fauna and fungi so often, or funga, so often are left out, despite their very long-standing separateness from plants and animals, and despite their great importance to ecology. So this for me, as someone who's been in the Senate and understands, you know, how governments work and government departments do things, I realise the great importance of getting the name in there to draw people's attention to the fact that they are important and that we know far too little about them and what they do. So just a little bit more about the group that decomposes, what they do, breaking down dead organic material, cellulose, lignin, chitin, using enzymes. So this is for the most part what fungi we have at Westgate Park doing this. And part of the reason for that is that Westgate Park has had now several layers, if I can say it that way, of wood chips and chopped up trees, basically. And so the predominance of species in the park are decomposers, and they like very deep mulch. So that's the case for top right there, the dung cup, Pizziza fasciculosa, that enjoys a very thick layer of mulch. And of course, what that's doing is reducing the accumulation of plant and organic material, building healthy soil, etc. Then there's the mutualists, the ones that have beneficial relationships with plants, and that turns out to be around 95% of plants. So there's an exchange of water, sugars, and other nutrients through the plant roots. There are this arrangement, it appears to be essential for healthy native plants and ecosystems, especially in Australia's impoverished soils. And there, of course, you know, people are astounded when I say that there are some fungi which actually penetrate not just the roots of plants, but invade plant cells. That whole concept that this could be possible with two separate organisms. And the parasites, of course, as with plants, fungi can be parasitic, but that also is a role which is important in nature in keeping populations healthy by weeding out the weak. And there are some examples of some parasitic fungi. And then the network. Networking and reproduction of fungi is just as fascinating, I think. What we can't see, of course, are all of the small threads of hyphae that form mycelial systems and ramify through the substrate. So just that very concept that the ground that you're walking on, perhaps not what you're walking on, but what you see underneath there is this amazing network of and structure of hyphae. Interestingly, not all fungi produce sporing bodies, and some are very small. Spores can be largely microscopic and various colours and highly individual. If you look under a microscope, you see what's called the decoration on spores, and that can be, in some cases, the only way in which fungi can be identified. Although now, with genetic sequencing, we are able to understand that much better. And here's a little illustration of how it goes around and comes around and under. All right, so getting back to urban parks, I can't recall any council document or state government document or even federal document that doesn't say that at the heart of what it's doing on the environment is encouraging biodiversity. And yet, I see examples of this all the time. Very rarely does fungi get a mention, and very rarely does is there a consideration of locally indigenous species and how they facilitate biodiversity? So, my thing, if you like, is to encourage greater local indigenous species in parks. So often our parks have lots of, lawn, imported species, lots of tall trees, and not much by way of in between. So we also don't have the right mix of structures. So we should have trees and shrubs and ground covers and lilies and sedges and grasses and so forth. This is what Westgate Park has done, and I think it's the reason that we now have such biodiversity. I'll get onto what our biodiversity in the park is in a moment, but I'm very convinced that this is an important element of biodiversity. You can't have true biodiversity without it. Some will argue with me, I know, but from observation of parks in my own area, there's very little by way of biodiversity if you don't have native species. What we have done at Westgate Park is establish the trees and shrubs first, so you give protection to the soil. Mulch is needed, minimise soil disturbance and compression, leave logs and branches and leaf litter on the ground, and avoid fertiliser, fungicide and herbicides. Now that's particularly difficult right now because we've had a lot of rain, but it's been very, it hasn't gone deep into the soil. It's been light showers more than anything, which is wonderful for weeds. And of course, because we've been locked out of the park during lockdown, we haven't been able to do much about them. So sometimes these criteria can be difficult to follow. This is a map of Westgate Park. So as you can see, it's very disjointed. You can see where the freeway goes through the park. So there is a space underneath the freeway that connects the park. The park is 2/3 in water bodies, so big freshwater lake, a saltwater lake which turns pink because it is extremely hypersaline. And so when the conditions are right, such as very warm weather, a lot of sunlight, then there is an interaction between, we think, an algae and a bacterium that produces what is a relatively natural, although unusual, phenomenon. So it didn't go pink in the last summer, but it had done 4 or 5 times prior to that. Okay, so the river goes along the top left-hand corner. So that's a river bank. We have the glorious vista of almost a kilometre of Yarra River frontage very close to the sea. So just down here in the bottom right corner, that's where the foreshore is, a bit further on from there. So you can see it's quite a sandy area there. That's where our Coast Banksia woodlands is. And so we have 9 what we call plant communities. They're not exactly EVCs, but they're plant communities, and they are different from one place to another. So this was determined by the Friends of Westgate Park very early on in the process. It was partly governed by the soil. What happened when the park was formed was that enormous numbers of truckloads of rubble basically were brought to the site to form hills and lakes and so forth. And that actually funded the work. So tipping fees funded the work. So there's quite a lot of concrete and bricks and so forth, not too far down. So we're very careful when we plant, but this was a highly degraded site, and now you would hardly know that. Okay, so that's just to give you the picture of where it is. Lots of sections. This, if you can see my indicated that area there was added recently, as was this reserve down here, previously a very weedy space. So now it has been revegetated, and we saw fungi in this area right from almost the beginning. So I doubt it was there under all the thistles and the blackberries, but it turned up somehow. All right, that's what it looked like during construction. So the rubble is now covered with a very light layer of topsoil. You can see the city. This is 1985, so the city is not quite as tall as it is now, of course, but you had good views straight across there. And so that landscape was planted out loosely with indigenous species. So they're mostly from WA, because that's where you get the really bright red species and the interesting plants. But it was then left pretty much and neglected. So the garden beds just became covered in weeds, and so they were mown. And in 1999, the Friends of Westgate Park started revegetation. It was actually discovered by a young woman. It's a very long story, so I won't tell it. But she was doing a walk down the Yarra River, arrived at the park, and said this could do with a lot of work, and I wonder who's looking after it. Anyway, she started up the Friends of Westgate Park, and 22 years later, you will see the result. So this is just a little snapshot of different parts of the park. So that's our river frontage, and as you can see in the background in the top left, the Port of Melbourne up here with cranes and so forth is present. The rock wall across here was completely bare. There weren't any trees in the background, so that was all planted out by us. In the top right, there is a new wetland, a series of ponds that it was, as I said previously, land that had lots of weeds. Bottom left is just a little example of the regeneration that we're now seeing. So after lots of application of mulch over many years, we have now stopped that, and we're seeing a more natural surface emerging. The topsoil's been built up sufficient to sustain plants, and so we're seeing regeneration at last. And that's just a little example of one end of our freshwater lake, and the little windmill which moves water around. So this is a typical bit of landscape, and you can see the logs on the ground. This was actually a pathway that was filled in. We had paths everywhere, paths and roads all through the park. So decided to consolidate and to create a bit more space for plants. So the Westgate Park biodiversity at the present time is 70, as Susie said, 70 identified species of fungi. So that's the various categories we've just been through. We've also had 4 different slime molds, and I know that slime molds are not fungi. If you were at the previous webinar, you would have heard all about slime molds, but I was delighted to see them in the park. And we've got 1, maybe 2 species of lichen. I have included right at the end of this presentation some lichen from elsewhere for people who are interested in that, but it's not from the park. We do have 168 bird species recorded over the last 18 years, so fabulous database of records there. We've got reptiles, long-necked turtle. They breed in the park every year. It's quite alarming to see their hatchlings gobbled up by birds, but that's nature, and that's part of our food chain, if you like. We have tiger snakes, we have blue tongues and other lizards and skinks. Flying foxes, obviously, microbats, absolutely. We're keen to start a citizen science program on what we've got. We see evidence of Ricali, though we haven't seen them, and we've seen loads of ringtail possums and frogs galore. We've only got 2 species, but after it rains, it's pandemonium. And then beetles, bugs, and butterflies, and moths, and damsels, and so forth. If you want to see them, they're on our website. That hasn't been systematic, though, and we're going to improve our efforts there. All right, so now we've got you know, a fairly natural floor, and we start to see some fungi popping up there that look a little different from what was coming up through mulch. And there we have it, Mycena viscidocruenta, I think that's how you pronounce it, ruby bonnet. I was delighted when I saw that. A very special little Mycena, I think, entirely red, as you would know, perhaps. And then Gymnopilus junonis. This is, sorry, I'll go to the next one, I think. No, we're getting into fungi now. Should I have stopped, I think, Ros, for a break? Ros?
Yes, this is a good time. We have some questions already. I've got 3 questions to put to you.
All right, good.
So you have a little break and I will. So our first question came from Michael Perkle, and he said, would you like to comment a little bit more on the Wood Wide Web?
Wood Wide Web?
Yes.
I'm not sure.
Or would you like me to comment on it for you?
Sorry?
Or would you like me to say a little bit about it?
Yes, why don't you?
Yes, yes. So the Wood Wide Web is a concept that was started off in Canada by a lady called Simone, I can't remember her other name, but she basically found out that the trees were actually talking to the other plants and communicating through the fungi from plant to plant. And so it was a case of trees caring for each other and caring for young trees and everything through. It's almost like the internet of fungi. So it's a lot more than just exchanging what we thought was exchanging water and exchanging minerals for sugars. It looks like there's a lot more exchange as it goes on, and plants even warn each other when there are predators around. And it's still a field that we don't know terribly much about. We're learning a lot more about it. And if you go onto the web itself, the internet, you can actually see there's quite a lot of information about what we're now calling the Wood Wide Web. So it's www. But it makes people much more aware of how important.
Indeed.
How important.
I'm sorry, I
just got a message to turn my video on, so I've just I tried to do it, but it didn't work. No. Oh yes. Okay. It looks like I'm on now. Okay. So so the second question is from Tash, saying, what can we do to actually advocate for government and local governments to recognise fungi so that we can actually get them protected along with flora and fauna? So Lynn, that's a question for you.
Yeah, well, that is the big question. And our advocacy, I think, needs to be kept up. We've been a bit limited in the last 12 months or so because of other considerations, but we need to get back to reminding government of this. And as individuals, you can do this as well, especially when, you know, major documents like the State of the Environment come out. So it's important to just keep asking the question, why is it that fungi is not being recognised for its importance to ecology? It's a tricky one because most of the research which is funded around fungi is for pest species. So it's agriculture. It's how do you get rid of the, you know, rusts and so forth in crops. So that's the challenge. A lot of the perception is that fungi is a problem, and not that it's actually useful, very useful. So I think, you know, we need knowledge. We need people to remind people who do know. We need them to remind those that don't appear to, that this is the case.
One of the very useful things you can do, certainly in Western Australia, a lot of our local councils have bush care officers, and they do things looking after their various bits of bush. And you can actually have a meeting with one of those, or go out in the field to your local park with them and talk to them about, you know, are you aware of the importance of fungi? And sometimes they are, and they're delighted that you want to know more about it. And they will often say, well, can you write something into my council so that I have some support from you, because the council doesn't even seem to recognise that fungi exist. So sometimes they don't know much and want to know more. Sometimes they know a fair bit and actually want your support. So going to your local council and your local bush care officer is really starting off at the grassroots level. And I think we need to come down from the top and up from the grassroots and try and sandwich them in the middle and make them listen.
Indeed. And one of the difficulties we face is the sort of siloed approach to nature. So we talk about groups separately as if there wasn't an interaction. And I think, you know, that for me has been a real, important realization that things are very connected in nature, and they have evolved over so long in being highly specialized as well. So, you know, it was all new to me.
But it's a great way to learn, and they have evolved to be together. They really don't want to be separated. Okay, the third question that I've got is from Susie, which is actually asking at Westgate Park, where was the place that you noticed fungi first appearing, and is there somewhere that fungi haven't appeared yet, or that they have only recently appeared?
No, look, I think when I noticed fungi at first, it was everywhere, so I couldn't say that. But I can say that in the areas where the planting first started, if you like, is where we get the most interesting fungi. So the cups, they're there immediately once the mulch goes down. The stinkhorns, same thing. But the mycenas, for instance, I've only seen on in areas that haven't been disturbed in a very long time. So yes, my favourite section of the park is called Woodland Walk. If you're going to the park, this is where you see the interesting things. And of course, a lot of them are tiny, so you have to stop and stare and look carefully. I still find things that I know the park intimately, and yet I still find places I haven't been before, and I'm quite sure that there would be fungi there. There are other observations. They're just casual observations, and I'd love them to be more scientific than this. But there are parts of the park where I only see one kind of fungi and it's not elsewhere. There are times, like a few weeks ago, when I discovered a little puffball I hadn't seen before. It was one that was on a stalk. And here was a whole group of them along next to a path. So I find new things all the time. I think I'm pretty observant, and so, or maybe I'm not, I don't know, but it's interesting to see that there are some sort of the good thing is being able to go to the park all the time. So if I probably go to the park at least once a week, and that allows you to observe when things are coming, see what's coming up together, see what never comes up over there but is here. But my answer is that it's at this stage, it's in my head. It's not part of any study, but it ought to be, and I hope we can move to that at some stage.
And it's also good to write down your observations, like, every now and then, because you'll find that when you look back over your notes, you're going, oh, because even your observations are valid. There at least something's happening.
Well, I record, so I take my camera. Whenever I go to the park, I take my camera, and whilst I don't take photographs of everything, I use my camera and its dating mechanisms and naming and so on to be able to be a record. So I did actually keep a record of where I saw what, and obviously I've loaded up observations to Atlas of the Living Australia. In fact, I sent them an enormous set of photographs from the park at one point, and I keep that up more or less.
That's excellent, and never underestimate your photos, because your photos are actually a very valuable record.
Yes, well,
Yep, there's no more questions, but there is a comment from Bev and Jenny saying that imported mulch is usually the major source of fungi unless soil is brought in when the plants are planted. But of course, we do have spores floating in the air as well. But imported mulch is food as well as containing spores already. So that's just a comment.
Yes, thank you. A lot of people ask that question, where have they come from? And the answer is, I have no idea.
A lot of them are floated in on the air. Yep.
Yeah, and I'm sure that they're up in the stratosphere because there is, you know, we share the same species with other parts of the world, so that that's likely to be the case. But, you know, I know, for instance, because stinkhorns, the method of spore dispersal is for insects to walk off with the sticky spore mass, and so they don't go very far. But there are other species that are everywhere in the park, not just in one location.
That's right. Okay, right. So we're ready for you to go back to your talk now. Thanks, Lynn.
Look, this is just a series of pictures with kind of a bit of a story with some of them. So there are in the park, and this is one species I'm not sure about. I probably got this entirely wrong, but we do have mushroom-like fungi, and but more commonly we have this beast. So Agaricus anthodermis is everywhere. I warn people about not picking it. Mostly people want to destroy little mushrooms they see like this, but I warn people that if you see them, they may look like they're edible mushrooms, but for the most part, they're not in the park. So the flat top, rub the edge of the stiper, and you'll get a yellow colour coming through, and you'll also have a quite sulphurish sort of aroma. This lovely little Springfield cap I haven't seen for a couple of years. That's the thing. I'm seeing differences. So it's not like the park is full of fungi every year. We do have to have really good rainfall, and the soil, even now, after the rain we've had, is still dry underneath. And so I think that's why we're not seeing any just yet. It may come further down the track. One of my favourites, and you will see this tiny little fungi. It's the same species here, so it's a little Bulbitius titubans, and it comes out of the ground with a shiny sticky cap like that, and very yellow. It's almost always set upon by colembula, tiny little black insects, jumping insects that proceed to start eating it. So it has a very short lifespan, and the colembula know that. So in the morning it can look like this, a little sticky ball, and then by the end of the day the cap is spread and becomes silvery and shiny, and then it kind of disappears quickly. So that's mostly in grass. What you can see here is that it's in mulch, but typically it's in the middle of the oval amongst the grass. We get a lot of Chlorophyllum burnium. I think that's how you say it. I'm sorry if I get the pronunciation wrong, correct me. So it's a very attractive mushroom and quite large, but it too should not be eaten. I don't know what species this is, but it's a very handsome little mushroom. And I think it's a clitocybe, but that's one that I'm a little doubtful of. There are lots of Copronellas in the park, so they come up quickly. They don't last very long. I actually have these in my back garden too, and the big native slugs come out at night and eat them. More Copronellas, and look, I think that's Lagopus, I'm not sure. And then these are much loved in the park because they're in prominent places, so they're the most often commented on, and they are, of course, ink caps. So they might look like on the left-hand side in the morning, and by the afternoon they're dripping their black ink and disappear. And this is a very beautiful little fungi, Gymnopilus delepus, I think is this one. And on the right-hand side is a massive log. So this log is semi-submerged in the soil, and it's been there for a very long time, so well before I first started coming to the park 10 years ago. But every year it produces spore bodies, and slowly this enormous log is being decomposed. It's the best example I can think of. And the other day I was there, and there were a few scrappy caps around, but for the most part the grass is now starting to grow through the log, meaning that it's almost done its job. So there's a kind of tail there. And this delightful little, I think it's a gymnopus. Again, you can correct me if I'm wrong. I love the way the grouping of these caps and the shape of the stiper and so on. It seems to me to be sort of eloquent and sinuous. I just think they are some of the most beautiful creatures. And then this one, I've only seen this on one occasion. And I understand it to be Lentionellus vinulus. And I left this one in because sometimes you don't want to disturb, in fact, for the most part, you don't want to disturb what's there, especially if you've only got a few of them and you've never seen them before. You want to leave them in place so that the spores are spread and they come back. So I always take a little mirror so I can put underneath, and that tells you a lot about what it is. So strongly recommend mirrors rather than pulling things up. But this one's been pulled up because there's lots of them. So I think this is Lepiota, but I'm not sure, but it's also very beautiful. And this looks like a lot of things. And it's a bit of a guess, really, whether this is Lepista or not. But it's pink. It's very pink. And on one occasion, we saw this wood bluet, Lepista nuda. I'm pretty sure that's what this is. I didn't get a very good photograph because it was so jammed up against a plant. But nonetheless, you can see that it's nice and purple. This is a really common fungi in the park, and again, it looks completely different depending on when you see it. So the little gorgeous little shiny red caps, the sort of elegant structure, the very long stipe, and that keeps changing names like most things. This is, I think this is what this is, and it. This is the one that only occurs in one little ridge in the park. It's a fairly sheltered spot, so that might be why it's there. But, I haven't seen it so far this year, but it's normally there. It lasts for quite a while. Quite a few Mycena. These ones emerging out of a log. I love I don't know what it is, but this little down the bottom corner, bottom right corner, I discovered this on a piece of bark underneath a eucalypt, a tiny, tiny, tiny Mycena. And if you look very closely to it, you can't see it on this photograph, but it's got a sort of a sparkly white cap, almost jewel-like. Astounding. But I've only seen that twice. The rooting shank, that's just changed its name recently as well, Hymenopellis radicata, and that has a shiny brown cap. It's quite easy to distinguish. I think I'm pretty confident about that one. And lots of these in the park. I think they're Parasola conopilus, and they start with this gorgeous little orangey red cap, and then as the caps dry, they turn this yellow colour. Loads of them around. Parasola plicatus, again, this looks very different when you see it as an emerging cap to when it opens up. I think that's Satharella. Again, amazingly slender stipe on these. Silicide we've got. I don't publicise where they are in the park. And then, of course, the delightful Schizophilum communis split gill. So haven't seen that this year or for a couple of years, but it seems to like to inhabit the stumps of trees that have been cut. So that's where I first saw it. And Tuberia ferferacea, another delightful looking, beautiful mushroom. This one is also likes a lot of mulch. It can push its way up through very deep mulch, and it's quite a sturdy mushroom, so it hangs around for quite a long time. Sometimes has a grey shiny cap surface. And this was quite special. This has only been in the park for the last 3 years, and our very first coral. So I was very excited to see this. It's tiny and looks a bit like a cauliflower sitting in the ground, but that's the green staining coral. So we're delighted to have that on board. And if it gets bruised, that's the colour it turns, this greeny colour. And the stinkhorn. So Clathrus ruber, red cage stinkhorn. We've got that in lots of places. So it starts with big bunches of that kind of egg-like structure. I'm told in the natural environment that these are relatively rare, but we've got loads of them. And its kind of partner, the Azarorubra, anemone stinkhorn, that's clearly liked a lot by small animals, so it gets eaten quickly. And bird's nest fungi, these are amazing, I think. So that little cap pops off. The spore dispersal of these is extraordinary. Each of those little petioles, I think they're called, is attached to the base of the cup by a string, and it waits for raindrops to hit the side of the cup, and one at a time pretty much these little petioles are sprung out of the cup and they land a little distance away. So you can just see a couple of them. I think there's one over there. So if you look very carefully, they're very tiny, but if you look very carefully, you can see how they're actually spread. And we have quite a few puffballs. This is like a perdon, I think. This one is very common, and we know that Scleroderma cepa is mycorrhizal fungi. So typically it grows on the edge of the path. I don't know why, possibly because it needs to be exposed in order to disperse its spores. It needs to be somewhere where it's relatively windy. Maybe it's there because there's a water runoff to the edge. Can't be sure. And then there are the earth stars. We've got lots of these. They're just delightful. Whenever I go on take people on a fungi foray, they just become so delighted to see them. They're pretty. They're, star-shaped. The amazing way in which they push themselves up above the ground in order to be able to disperse their spores. The whole idea that they're waiting for a drop of rain on that quite papery little spherical part, so that a little puff of spores comes out. I often show that to children and they're just delighted by it. And then you can always see Pycnoporus coccinaeus in the park. It seems to like paper bark trees, and we have lots of those. We cut them down mostly because they're woody and weedy. And so they're lying about and they get, this is a polypore. So it doesn't have gills. It has these little kind of looking like drilled holes, and it has this beautiful bright orange colour until it gets algae all over it and goes green. Another really common fungus is Trimetis. You'll be familiar with that. Comes in all sort of shapes and colours, and it's also spores. These ones I don't know what they are, but I thought it was worth putting in there because they're pretty interesting. And here are our cups. So for the most, I think we've had one variation, one different species, but I think these are all Pisiza fasciculosa, the common dung cup. And what I love about them is they're translucent cups. So if you get sun striking them in a particular way, you get this lovely glow, this lovely orange glow. They don't seem to be eaten by anything, so they're usually pretty intact. And then the slime molds, the amazing slime molds. So I won't go into this, we're probably running out of time, but just to show you that we've got them. Leocarpus fragilis, which has these beautiful shiny little balls of orange that go brown, and then that becomes the spore mass. And then Fuligo septica. So we have this in the park, and that's just the good thing about being able to go back and back and back, you know, is that you can record what happens over time. So this is the first stage where it looks a bit like scrambled eggs, and then it goes a pinkish colour. You'll see the little, I'm not sure what that's called. It wouldn't be hyphae, but it might be something else. But this moves. It moves over the ground, and as I understand it, it consumes bacteria as it goes. And then it dries out, and this is the final form of it, which is just a great big spore mass. And this is clearly a slime mold, but I haven't been able to find it anywhere in any textbook, but I love it because it reminds me of jumping little men. I've got, I don't know if people are interested. How are we going for time? Should I stop here, Roz?
Yeah. Just do about another 5 minutes. You've got a bit more time, and then we'll have a bit of time for questions.
Okay, all right. So look, I'll just fly through this. I became more interested in lichen, which of course is a combination of a fungus and a photosynthetic partner, photobiont. And so I accompanied Sapphire when we went to a workshop in northern Victoria, and we found this lovely coral lichen. And this one, I mean, interesting that there are so many of them around, 30,000 species covering 8% of the land. I'm also in the Dry Stone Walls Association, and I'm much more interested in the lichen that grows on the stone than I am in the wall construction, I have to admit. I find them absolutely fascinating. This is a desert species, not a very good photo. It's a bit fuzzy, but that's a very common one. And so there are three main groups I discovered. There's the fruticose, the foliose, and the crustose. Roughly speaking, they fall into those categories. Some of them are just gorgeous. This is a ramalina of some sort. And there you can see how this is the fungi, which is the partner, is most often, as I understand it, a cup, the cup that we've just been looking at. So that shows you little cups, and that's where the spores are dispersed from. It's not unusual to see on one branch as many species as this. This is up in the high country, so I was able to identify what lichen that was. This one is much more common. People have seen that. It's for Flavoparmelia. This is if I thought fungi was difficult to identify, this lichen is much more devilishly difficult. And this one was in the park, so nice little Xanthorea parienta. Okay, so I'll just quickly flick through these. This was from up in the mountains to Cladonia with a little trumpet. So its cup is just at the top of a kind of tube. Pretty. And you'll be familiar with the Usnea. Anywhere you go, you see them. And just one. And then the various crustose lichen. So this is what I see when I look at dry stone walls. That's just a little image of Alison Pulio and a workshop she was doing, and a reminder to use iNaturalist, and a nice little, graphic down there. And then this is just to remind you that FungiMap has a terrific shop with lots of wonderful publications. Okay, so I suggest that you, if you haven't already, I'm sure you have, but if you're not on eNews, join, register for that, join FungiMap, and look at our website for all those things. I think that's it, Susie.
Okay, well, I have just two more questions at the moment for you to answer, Lynn. So the first one is, in Westgate Park, have you noticed any fungi along the sandy banks of the river that might not occur elsewhere?
We do have a sandy bank, but it's mostly underwater along its length. So the rock wall has been put there, so we don't have a dry sandy area as such. So no, but you know, we talk about fungi being in every ecosystem and everywhere, but I think there needs to be more work done on that so we can know what to look for. I haven't even seen any images of fungi that's underwater. I gather it's there, but no, I can't say I've seen any at Westgate Park.
Fair enough. And the other question is, do you have any idea why there's only a few species of lichen in the park yet? But then Susie's also answered the question in a way, are the trees and rocks not old enough yet, which is possible?
I think it's because there's so much pollution in inner Melbourne. We're talking about a park which is surrounded by industry. So it's got the port on one side, it's got a printing organisation for newspapers. It's got Boeing. It's completely surrounded by freeways, by roads, by all of that. So, as I understand it, lichen doesn't much like a polluted atmosphere, and so so that might be the reason. Although I've noticed in recent times, I live in Port Melbourne, and I've noticed that the tree in front of my house is now sporting lichen. So maybe we're cleaning up our air quality. I don't know, but that's, as I understand it, a very important factor.
One of the studies that was done, I think the 1800s, was actually noticing in England that lichens were disappearing as pollution was getting worse. So some very early studies were done actually on lichens.
They seem to be very sensitive to,
very sensitive,
well, like the canary in the cage, really.
Yes, that's certainly the case. So there's another question about what is the size of Westgate Park?
40 hectares.
Right, so that's, yeah.
It's a good size, and it's big enough to sustain, I think, the biodiversity. The fact that we have, over 100 species of birds recorded in the park suggests that it's big enough for them to be far enough away from people.
Yes.
So Westgate Park is not heavily populated by people. It will be into the future because there's a massive development with Fisherman's Bend right next door. But I hope we can maintain the bush-like nature of the park so that we so that we can keep all those animals and fungi still going there.
And it's obviously what people value as well.
Well, I think so. I think so. They certainly value, when we do fungi forays, it's filled like that. There are many more people want to do it than we can accommodate. So
and that moves on to the next question, which is, when will you be doing a fungi foray?
Oh, I wish I knew.
Yes.
The problem is to get the timing right. Often you need to give people notice and warning, preferably 2 weeks in advance. But of course, you need to be there the day after the rain, unless it's been very, very wet for a long period of time. So we may or may not do a fungi foray even this year. There was one year when we could have done them continuously, but it does unfortunately require rain.
Yes, it certainly does. Well, thank you. That's all the questions that we've got at the moment. So thank you very much. Oh, there was actually one comment on the other one, which was about, has the park actually ever thought about putting in commercial fungi as well? And one of the things I would like to say to that is, like, we know what happened when we brought rabbits and cane toads and things like that. Don't bring in fungi from other countries. And most of those commercial fungi are fungi from other countries. So we don't want to do that.
Yes, there was a great interest in bringing a fungi into the park that might be mycorrhizal, so that because we're sure some of the plants we put in just don't survive. They should do, but they don't. Yeah. And we've concluded that it's because they don't have a fungal partner. So we did, explore this question, but it's more complex than simply getting a bag of spores from, the United States. I think typically where you get them from. All that does, I'm told, is encourage, it benefits the weeds rather than the specific species that you might have there. So that's a pointless exercise. In fact, it's more likely to be damaging. But we would like, at some stage when we have enough money, we would like to be able to sample the soil to see what's missing. So to go into a natural environment and to sample the soil for a particular species of plant and see what fungal partners are there, so that we can then compare that with the soil that we have in Westgate Park. I'm guessing our soil is nowhere near the maturity and the sort of stability, if that's right, to be able to see those interactions at this point in time. It may be coming already for some plants, we just don't know, but it would be terrific to do that study.
It would be, and as DNA work increases and the things that we can do with DNA, it will become easier to be able to do this
and
stuff.
Cheaper, hopefully.
Yes,
it's still quite expensive to get sequencing done, as I understand it.
The price is actually starting to come down, but it is still reasonably expensive.
Yeah, like renewable energy.
Exactly, that's right. We want that too. Okay, Susie, I'll hand over to you now.
Thank you, Ros. Thank you, Lynn. That was great. Really good insight, I think, for me. I could see into the progression of growth of fungi in Westgate Park. So an interesting observation, I suppose, of regeneration. And as you said, no fungi was deliberately brought in, but then bark and chip was brought in. So kind of it was in one way, but that's okay. We can all live with that. For those of you who still have questions, please email them attention to Sophie at fungimapcoord@gmail.com, and we'll endeavour to answer these questions. And pardon me, Lynn will supply them in due course. Hi, Soph. If you'd like to join FungiMap, purchase any books, or just have a look, please go to our website, fungimap.org.au. Also, if you've enjoyed tonight's webinar with Lynn Allison, you may like to book next month. I don't have the date confirmed yet, but Dr. Susan Nuski, it will be a daytime one at 11 a.m. She did a PhD study into the interaction between fungi, mammals, and plants. specifically the dispersal of ectomycorrhizal fungi by mammals in North Queensland, including the endangered northern bettong. Very interesting, and reason I asked some of the questions I did, pardon me, dying here, sorry, was that her space where the bettongs, where she worked and the bettongs are, is squashed. Sorry, it's between the grasslands and the sclerophyll forest. I'll stop talking. It should be very interesting. Keep an eye out. Thanks, everybody.
And
Good
the
night.
e-news will tell us when it's on. That's right. Thank you so much. Let's go and have a drink. Okay.
Sorry. Thank you, everybody.
Thank you very much, everybody, for attending. And thank you, Lynn, for a lovely presentation.
Thank you.